In the US, more than 100,000 people are waiting for organ transplants. Because of the organ shortage, around 20 people die each day. Mechanical or biomechanical organs can be partial or temporary measures of repair. Mechanical organs are synthesized from metals and inanimate polymers such as plastics, whereas biomechanical organs are composed of a combination of living cells, inanimate polymers, and metal. For end-stage organ failure, or the final stage where the organ can no longer function independently, biological organ transplants, made up only of living cells, biodegradable polymers, and metal elements, are the only treatments that can completely restore function. This treatment is done by patients undergoing procedures to receive organ donations from other people. This strategy presents a myriad of problems. Since it is not the patient's original organ, the body would generate an acute allograft rejection response. There are direct and indirect ways immune system T cells can be activated by the recognition of alloantigens. The direct method has CD4 plus T cells induced by the detection of major histocompatibility complex class II molecules. The indirect method detects the peptides of allogenic MHC molecules instead, while a semi-direct method exposes antigen-presenting cells of the host to MHC peptides through MHC transfer. These methods result in T cells being activated and proliferating from the recognition of alloantigens, producing three possible ways of organ rejection. One method produces IL-2, leading to the proliferation of cytotoxic CD8 plus T cells, increasing the immune response. A second method generates antibodies through the induction of B cells. Lastly, Fas/Fas ligand-mediated cytotoxicity, which can lead to apoptosis, will directly impair donor organ function. Thus, patients must take a lifelong supply of immunosuppressants. The side effects include an increased risk to cancer and infection. Compounding these issues is also the organ shortage. One solution to these problems is receiving transplants from animals. The question arises, which animal is the best to receive organ donations from? Many consider primates as great candidates due to their evolutionary similarities as well as their resistance to some human diseases like HIV and hepatitis B virus. However, the presence of monkey viruses poses a health risk for the human patients and those that come into contact with the primates, discouraging them as potential donors of organs. As of now, pigs are the best candidates. They have larger litters, shorter gestation periods, and are anatomically and physiologically similar to humans. The fear of endogenous retroviruses jumping to humans also raises concern. However, with CRISPR-Cas9, a technique to edit genes, pigs free from ERV have been created, preventing the transfer of the retrovirus. Another potential setback includes the potential rejection by patients due to antigen-antibody binding. It was observed that when human blood contacted pig organs, antibodies would bind to the alpha-1,3-galactose carbohydrate. This antigen-antibody interaction would reject the organ. Strategies to circumvent this issue were infusing competitive carbohydrates against the antibody or modifying the animals to remove or reduce alpha-1,3-galactose antigens. An alternative solution, however, is growing and harvesting organs in animals or in vivo. This technology was made possible due to the discovery of stem cells by Professor Shinya Yamanaka. Originally a doctor, Yamanaka's hospital experience and patient interaction inspired him to find ways to eradicate disease through preventative means by further understanding their mechanisms. When asked,
まず
why
25
did you
年
do
前
research
に
on
医
iPS
者
cells?
になり
Yamanaka
ました。整
responded,
形外科医にな
Firstly,
りました
25
が、
years ago, I became a
残
doctor.
念ながら
I
手術
became
が
a surgeon, but
あん
while
ま
it
り
was
上手じゃな
unfortunate
か
that I
っ
was
た
not
の
good at
で、
surgery, I
研
turned to
究
research.
者の道に転じました。
ES cells, or embryonic stem cells, originate from the blastocyst stage of the embryo. As pluripotent cells, they can develop into tissues originating from the three germ layers of the embryo. iPS cells are induced pluripotent stem cells, where adult somatic cells that have already differentiated to form their respective tissues have been reversed to become pluripotent like an ES cell. The research indicated that adult somatic cells could be de-differentiated, allowing it to potentially develop into something else instead. Technology originally achieved this in 2006 through a mouse in Yamanaka's lab in Kyoto. This discovery was then applied to humans in 2007. OCT3 and 4, SOX2, KLF4, and CMYC were introduced into HD SLC7A1. a cell that originated from the facial dermis of a woman. After being isolated and harvested, cells were allowed to grow. Colonies similar to human embryonic stem cells, HES, were found after 30 days. They continued to grow, developing into flat colonies. This development paralleled HES cells. In addition, like HES, these similar colonies also underwent spontaneous differentiation in the center and expressed HES markers like SSEA3 and SSEA4 and TRA160 and NANOG proteins, which were all cell-specific surface antigens. Researchers are hoping to apply the technology of iPS cells to grow organs for donation. They intend to do so by injecting iPS cells into a host embryo, where they will develop to the appropriate size for the recipient. This solution will circumvent the organ donation shortage and help prevent an allograft rejection. Hiromitsu Nakauchi's 2010 research observed that mice could host and grow a rat pancreas. During his further research with a colleague, they even observed successful cases of transplanting an insulin-producing mouse pancreas tissue into a diabetic mouse. However, it has proven to be much harder to grow iPS cells in larger and more genetically different organisms. Attempts of inserting human iPS cells into pig failed, where half of the fetuses had stunted development. Those that did grow to a normal size unfortunately only contained a few human cells, which was not sufficient enough for organ transplants. One of the fundamental issues is that human stem cells lose against those of the host. So, in a form of competition, the human stem cells do not survive. Furthermore, human or primate iPS cells are also considered more advanced or primed than those of rodents, which are viewed as naive. This feature reduces the likelihood of human cells surviving in a chimeric embryo. To define a chimera, it is an organism that has two genetically different cells. This quality can be advantageous for organ harvesting. If we manipulate the host embryo to not grow a particular organ, the injected donor stem cells would be the only components comprising the newly developing organ. For example, if we knock out PDX1, which is an essential regulatory gene for a pancreas, the zygotes would not be able to develop the organ. The supplementary stem cells, however, can then be introduced to develop the desired organ in the chimera. Such method is termed embryo complementation. One way of making such chimeras is through a process of blastocyst injection. This strategy injects the stem cells into the blastocyst cavity of the host, where it can then develop. The second strategy is aggregating embryos. Although it has a lower survival rate than blastocyst injection, its higher chimerism rate makes it an attractive strategy. Lastly, in utero transplanting injects the stem cells in the early development. According to the New York Times in 2021, a genetically engineered pig had grown a kidney compatible with the human body. At NYU Langone Health, surgeons attached the kidney to a brain-dead patient. The kidneys were connected to the blood vessels of the upper leg and functioned normally while outside of the abdomen. The successful procedure suggests that gene editing and transplants could be possible.